Today we'll be looking at Sphingosine 1 phosphate receptor modulator, an immunosuppressive for multiple sclerosis. Multiple sclerosis, MS, is a chronic autoimmune disorder characterized by neuron demyelination and neurodegeneration within the CNS produced through autoimmune inflammatory mechanisms. There are several forms of MS, with relapsing remitting MS affecting about 85% of patients. The hallmarks include lesions contributing to optic neuritis, loss of motor control, fatigue, and other neuropsychological issues such as depression and memory loss. Now, there are 5 main steps of drug development. 1. Target selection. 2. Lead discovery. 3. Preclinical development. 4. Clinical trials. And 5. Registration. So first, we look at target selection. Initially, the target for multiple sclerosis was unknown. The original objective was to find a drug suitable as an antibiotic, muriocin. This progressed on to inadvertently discovering that a derivative of muriocin was effective in treating autoimmune diseases, specifically MS. In 2002, Sphingosine 1 phosphate receptors were found to be effective in treating models of autoimmune disorders. The activation of these receptors caused an antagonizing apoptotic effect that were associated with the earlier stages of neurodegenerative and demyelinating diseases. Secondly, we look at lead discovery. Muriocin is an antifungal and immunosuppressive compound that was isolated from culture filtrates and mycelium of Miracoccum abomyces. This fungus was identified from the phenotypic screening of large amounts of thermophilic microorganisms for antibiotic activity. Muriocin was a potent immunosuppressive agent both in vitro and in vivo, appearing to be a candidate for clinical applications such as organ transplantations and autoimmune diseases. However, it was actually too toxic for therapeutic purposes after characterizing its physical, chemical, and biological properties. Then, in an attempt to create chemically modified compounds derived from muriocin in 1994, FTY-720, otherwise known as fingolimod, was identified. And at this stage of development, the immunosuppressive properties of fingolimod was actually unknown for the treatment of multiple sclerosis. In 1995, fingolimod was concluded to have a significant immunosuppressive activity, as well as improvement in side effects that muriocin otherwise had. It was theorized to be a safer candidate for organ transplants and autoimmune diseases. Next, we move on to preclinical trials. In preclinical trials 2004, FTY-720 was administered to mice with brain inflammation. This resulted in a sustained improvement in clinical score during administration and changes in mRNA expression encoding within some myelin proteins and inflammatory mediators. FTY-720's previously known activity is based on its ability to bind to Sphingosine 1 phosphate receptor 1 on circulating lymphocytes. This activity reversibly traps a proportion of the lymphocytes in the lymph nodes to reduce the number of inflammatory T cells circulating through the bloodstream and in the CNS. In 2007, another study conducted in cell culture demonstrated that FTY-720 improved the survival and increased the number of immature and mature oligodendrocytes, cells that help form myelin in the CNS. Stimulation of S1P receptors on the oligodendrocytes may help limit demyelination and/or promote myelin repair. A third study in 2008 was conducted on a rat model of MS, where researchers found that FTY-720 directly administered in the rat's brain significantly suppressed the severity of MS. This suggested that there was additional mechanisms of action that were independent of lymphocyte depletion. In imaging studies, enhanced myelination and axonal protection associated with clinical effects were confirmed in rats receiving oral FTY-720. Finally, in 2009, a study evaluated the ability of FTY-720 to activate S1P receptors on astrocytes in cell culture. This is because astrocytes play an active role in nervous tissue repair and to regulate the permeability of the blood-brain barrier. In this study, it was found that FTY-720 actually activates S1P receptors on astrocytes, stimulating intracellular pathways that regulate a variety of functions, such as cell proliferation and migration. Afterwards, we move on to clinical trials. In 2006, 281 patients received either a 1.25 mg or a 5 mg dose of fingolimod or a placebo once daily. The results show that fingolimod reduced the number of gadolinium-enhancing lesions detected by MRI and clinical disease activity in patients with multiple sclerosis. In 2010, 171 Japanese patients received an oral dose of either 0.5 mg fingolimod, 1.25 mg of fingolimod, or a placebo, all of which was taken once daily across a period of 6 months. The results showed that a high proportion of patients that took fingolimod were actually free from gadolinium-enhanced lesions at months 3 and 6. Fingolimod reduced lesions better than any placebo, but not significantly. These results for Japanese patients were also consistent with Caucasian patients later down the road. In 2011, 1,083 patients received an oral dose of 0.5 mg fingolimod, 1.25 mg fingolimod, or a placebo, all of which was taken once daily across 24 months. The results show that it was beneficial as a disease-modifying agent in the management of multiple sclerosis. In 2010, another study had 1,153 patients receiving an oral fingolimod dose of 0.5 mg and 1.25 mg daily, or a weekly intramuscular interferon beta-1a dosage of 30 micrograms across 12 months. The results showed a superior efficacy of oral fingolimod with respect to relapse rates and MRI outcomes in patients with MS, compared to that of intramuscular interferon beta-1a. Any other information on other phase 3 clinical trials are found in the last slide. Finally, we come to phase 4 trials, 2017 to present. In these phase 4 trials by Novartis, more than 75% of patients treated with the drug had either no relapses, no new or enlarged MRI lesions, or no progression of disability over a period of 16 months. In addition to this, 58.2% of patients had no MS-related brain shrinkage over a size of 4%. This is the broad range that people without MS would otherwise experience. Lastly and finally, we move on to registration. After these extensively tedious processes, in September 2010, the FDA approved fingolimod for the use of relapse-onset multiple sclerosis. Later, it was approved for use in Australia by the TGA in January of 2011. So, in summary, the development of fingolimod for MS did not follow the traditional drug development and design process. Myriosin, the natural isoform of fingolimod in Myricoccum albumicines, was first isolated without MS as a target. Myriosin was initially deemed too toxic and modified to produce FTY720, which was later found to be useful for MS after discovering its immunosuppressive properties. After the successful preclinical and clinical trials, fingolimod was registered to be used by the FDA in 2010 and the TGA in 2011. And with that said, this concludes our Sphingosine 1 phosphate receptor modulator timeline.